Hello, everybody. Welcome to session 2 of Unit 7 Developmental Biology. And today we'll be discussing development of frog. So, the embryonic development of frog it is completed with the following sequence. There are total 6 stages. The first one is fertilization, cleavage, morulation, blastulation, gastrulation, and lastly organogenesis. So whenever you explain it, we should explain in sequence, in serial manner. So now let's begin with how the fertilization takes place during the development of frog. So fertilization, it is the process of union of ovum and sperm, and there are two types of fertilization. One is external and other is internal. So in amphibian, since frog is amphibian, so in frog the fertilization is external because the union of sperm and egg it takes place in water outside the female body. So during the fertilization, male frog it produces a kind of sound that is called croaking sound by the vocal sac, and that sound is produced to attract female frog for mating purpose. And when that sound is heard by the female frog, then the female frog goes nearby the male frog, and male frog mounts over the female frog, and with the help of the forelimb, which has got copulatory pad, it will grasp the belly of the female frog firmly. So due to the pressure of forelimb of male frog, uh, the, the, the eggs which are stored in the belly of the female frog, it will be released outside. So this mating, actually it is not the real mating, just the male frog mounts over the female frog, and since male frog doesn't have penis, so that is why there will be no actual sexual interaction takes place. So here the male frog mounts over the female frog and releases the uh, eggs by the female frog, followed by spray of uh, sperm over it by the male frog. So this kind of the mounting is known as amplexus or pseudo copulation. Here pseudo means false, copulation means sexual interaction. So here there is no any genuine sexual intercourse, that's why it is called pseudo copulation, or it is also known as amplexus. So once the female lays the eggs on the water, then the male frog will spray the sperm on it, and this process is known as insemination. And let me tell you, when the female frog lays the eggs, the egg is not completely matured. It is laid in the form of secondary oocyte that we discussed during the gametogenesis. So the female frog lays the egg in the stage of secondary oocyte with one polar body. And once the sperm is uh, entered into the egg, so the head of the egg and the head of the sperm has got the special modified Golgi bodies. So it produces the enzyme, and that because of that enzyme, it can penetrate through the vital membrane and the plasma membrane, that is the inner layer. And once it is entered, then only the sperm, the head and the middle region will enter, but tail region of the sperm will remain outside. So once the sperm penetrate completely inside the egg, then the vitelline membrane it will come to the normal condition, whereas the plasma membrane still has got pores from where the albumin will come outside and form the thick and impermeable membrane, and that is called fertilization membrane. And because of that, the remaining sperms cannot enter inside the egg to avoid polyspermic condition. Now finally, the nucleus of both sperm and the ovum, they will fertilize together, they fuse together and form the diploid zygote. After the formation of zygote, um, about 2 to 3 hours later of fertilization, the zygote will undergo repeated mitosis cell division and ultimately form the multicellular embryo. So the division of zygote is holoblastic because here the division takes place completely from animal pole to vegetal pole, and ultimately at the end you can find two kind of cells. Small cells are called micromeres and the larger cells are called megameres. That's why the division of the zygote is holoblastic and unequal. The first division is vertical. You can see here, this is the first division. So here the division takes place vertically, starting from animal pole to vegetal pole, dividing two equal cells. And these two cells are equally divided, and these newly formed cells are called blastomeres. And this stage is known as two-celled stage because it has got two cells. After the first division, the second cleavage or second division occurs again vertically, but 90 degree to the previous division, forming the four equal cells or blastomeres, and this stage is known as four-celled stage. Third division is horizontal, and whenever it is dividing horizontally, then it will divide unequally, and the division will take place just above the equator line. This is the equator line. Just above the equator line, the third division takes place horizontally, forming unequal blastomeres. The upper four cells are small cells because it is divided above the equator line. So it will form the small cells, and those small cells are called micromeres. And the lower four cells are larger, and it is called megameres. Fourth division is vertical, resulting in each there will be the vertical division, and so the 8 cells structure will turn into the 16 cell stage. So here upper cells are the small cells called micromere, and the lower 8 cells, total 16 cell stage. So out of total 16 cell stage, upper 8 cells are small cells known as micromere, and the lower bigger 8 cells are known as megameres. And let me tell you, the upper cells all are lying in the animal pole, which are present with the albumin, and the lower megameres, they are present the yolk, which is having the yolk, and these megameres and micromeres, they grow further due to the nutrition provided by albumin in the upper side and the nutrition provided by yolk in the lower side. So after fourth division, the fifth division is again horizontal, and it occurs in two places. One is upside and other is lower side. So it is just happens on the either side of third division. This is the third division. So on the either side of third division, this is the upper one, this is the lower side. So it occurs in two areas. So due to which the 16 celled stage divides into from the 32 celled stage, where the upper area have 16 small cells called micromeres, and the lower area has got bigger cells called megameres, which are 16 in number. So in total 32 blastomeres are present in this fifth division. Now morulation. So after we just have discussed this now, that after the fifth division, the cells, the embryo will have 32 cells, and this condition, this looks like mulberry fruit. That's why it is also known as morula. And the process of formation of this morula is known as morulation. And here, out of 32 cells, the upper 16 cells, they they are micromere, and they are present in the animal pole of the egg. And the lower 16 cells, they are larger cells known as megameres, and they are present in the vegetal pole of the egg. So up to morulation, the cell division was happening in order. That means the division is regular. But once it enters into the blastulation stage, then there will be no any synchronization in cell division of the cells present in the embryo. So here the upper cells, that the cells present in the upper area, the upper area means animal pole, they are small in size known as micromere, and the cells present in the vegetal pole, they are larger in size known as megamere. So after the fifth division, the the cell division does not divide, does not occur in the similar manner. So they divide in different rate. So it means to say that micromere, due to small size, it divides rapidly in comparison to megamere. So because of that, when the micromere, as I told you, micromere divides faster than the megamere, due to which the number of megameres will be more compared to the number of megamere, due to which inside a cavity is formed, and this cavity is known as blastocyst, and the structure that you see here, this structure is known as blastula, and the process of formation of blastula is known as blastulation. Actually, this cavity, that is blastocell, it starts to form when it was in 8-cell stage during the embryogenesis. So this small cavity is known as segmentation, or it can be also called as the simply a central fluid, central cavity filled with the fluid, and that fluid is uh, that cavity is known as segmentation cavity, and it is filled with the fluid made up of uh, it is made up of albumin. So albuminous fluid that is present inside the blaster cell, that helps to develop the embryo further. When you check the fully formed blastula, then blaster cell, you can see in the figure, it is completely present in the animal pole. Whereas megamere, still you can see, which are the larger cells, they are present in the vegetative pole of the egg. So this is how the blastulation takes place. So the blastulation has occurred due to the rapid division of micromere compared to megamere, due to which the number of micromeres are large in number, forming the cavity inside, and this cavity is known as blastocell, and this structure is known as blastula, and the process of formation of blastula is known as blastulation. Now, when the blastulation is formed, then you have seen already that inside you can see the cavity known as blastocell. And when you look at the blastocell, then the roof of blastocell, it is made up of micromere, whereas the floor is made up of the megameres that you can see clearly. And the cells present in the blastula, these cells start now differentiating into different layers, namely ectoderm, mesoderm, and endoderm in future. And these areas later later divided into three layers: ectoderm, mesoderm, and endoderm. And the study has been done by the German embryologist Professor Vogt by using the technique known as intravitum staining technique. So he used the stain, and using that stain, he could study the presumptive areas of blastula. It means to say that in future, which cells turn into ectoderm, which cells turn into mesoderm, and which cells turn into endoderm. So let's discuss about it. Micromeres, which is present at the roof of blastula, it will form in future two structure: epidermis, this area, and neural plate. So the micromers which are present at the roof of the blastocell, in future it will form the epidermis and neural plate. So neural plate means later it will form the spinal cord and brain. Similarly, micromeres present at the dorsolateral side of the blastula, it gives the notochord. This is notochord, and lateral side of micromere, it will form mesoderm. It will form the mesoderm. So once again, the roof of blastula, it will give in future epidermis and neural plate, and dorsolateral side of micromere, that will give the notochord. This is notochord which will turn into spinal cord in future, and the micromeres which are present on the lateral side, it will give the future mesoderm. This is the mesoderm. And finally, the cells which are present in the vegetative pole, they are called megamere. They will form the endoderm. So there are total five different layers that you see here. So the roof will give epidermis and neural plate. The lateral side, dorsolateral side will give the notochord. This is the notochord, and lateral side of micromere will give mesoderm, and finally the megamere, which is lying in the vegetative pole, will give the endoderm. That means out of five layers, the upper four layers will be given by the micromeres, and the last one, the endoderm, will be given by megamere of embryo. Now let's go to the gastrulation. It is the very important stage of embryogenesis. So gastrulation, it is the process of formation of gastrula. So gastrula is three-layered embryonic stage, triploblastic we can say, and it is formed by the migration and rearrangement of cells present in the blastula. So gastrulation is a bit long process, and it occurs by following events. The first is epiboly, emboli, contraction of blastopore, involution, and rotation of embryo. So the first step is epiboly. So as I have shown the figure before, the upper part of blastula, it contains micromere, and the lower part of blastula contains megameres. And also I told you that micromere, it divides more repeatedly than the megameres present at the vegetative pole. And since micromere divide more repeatedly, so micromer number of cells will increase rapidly, due to which the micromeres will completely cover the vegetative pole. So vegetative pole will be completely covered by the micromere except the region of yolk bulk. So that I'll discuss later. So it will completely cover the embryo except the yolk bulk. So why micromere covers the whole embryo? Because as I said before, micromeres divide more repeatedly. So because of that, there will be formation of more micromeres. So that is why the micromeres will completely cover the embryo except yolk block. And this is called epiboly. Epi means upper and boly means of covering. So epiboly. Now emboli. So emboli is also known as intucking or invagination. So here, the small groove will form just below the gray crescent region. So gray crescent region I have discussed before in the topic egg. So after the fertilization, the place from where the sperm has entered, just opposite to the point from which the sperm enter into egg, you can get the gray crescent region, which is gray in color. So Just below the Great Crescent region, a small group appears due to invagination, due to the depression of micromeres present in this region. And this region is just below the Great Crescent region. And this depression is known as blastopore. And this blastopore is surrounded by four leaves. So upper leaf and ventral leaf and then two lateral leaves. So it is surrounded by the four leaves. So the group, it goes more deep inside. So you can see in the first step, you can see small group, but when it goes further development, then this group will grow more inside. It will sink more inside, forming a new cavity, and this cavity is known as Archenteron cavity, which will turn in future into alimentary canal. So here you can still see the blastocell. This is the blastocell that was formed during the process of blastulation. So once the Archenteron cavity is formed, then the size of the blastocell keeps on decreasing, and finally it will be disappeared, obliterated. And here the Archenteron size will go on increasing, and this Archenteron is opening outside through blastopore, and this blastopore is already, I told you, it is surrounded by the four leaves, dorsal leaf and ventral leaf and two lateral leaves. So total four leaves are present, and this blastopore in future it will turn into cloaca in the frog. So in frog it is turning into the cloaca, but future anus, but in invertebrates it will turn, the blastopore will turn into mouth. So if the blastopore turns into mouth, then this condition is known as protostomia. So it occurs in invertebrates, but in the case of frog, along with the other vertebrates, this blastopore will turn into the anus, and this process is known as deuterostomia. So deuterostomia means blastoma turning into anus, and protostomia means blastopore turning into mouth. And in vertebrates, the blastopore turns into anus, whereas in invertebrates, like in ortho, the blastopore will turns into mouth. So this condition is known as protostomia if mouth is formed. If anus is formed, that is called deuterostomia. Now, contraction of blastopore. So this is the third event of gastrulation. So here what will happen, the four leaves that is present in the blastopore, it start contraction. So they start contraction and form a ring-like structure, and that ring-like structure is called blastopore ring that you can see here. This is the blastopore ring. And due to the more and more contraction of these leaves, what will happen, the megamere which is present inside the embryo, it get the pressure, and because of that pressure, some of the megameres, large cell, they come out from the blastopore and form a kind of plug, and that plug is called yolk plug. Involution. So what will happen here is the mesoderm that is lying here, this mesoderm start to migrate inside. It will start to migrate inside and finally join with the notochord. And when it joins with the notochord, then this structure is known as chorda mesodermal layer, and this layer later forms the complete mesoderm. Now, rotation of embryo. So due to the arrangement and migration of cells, what will happen is that center of gravity of embryo it will change, and because of that, the embryo will rotate by 90 degree, so that animal pole, it will become the anterior part of the body embryo, and then blastopore, that future anus, that will become the posterior position, which will become parallel to the surface of water. And this is happening because the center of gravity of embryo changes due to migration and arrangement of cells. So Here what you can see is that when this process occurs, then what will happen, as we have seen in the previous figure, the size of the uh, blastula before it was whole blastocell was present. And what will happen once the depression will start, then the depression will sink more and more, forming the new cavity known as archenteron. So due to formation of archenteron, the size of the blastocell, it goes on decreasing. So you can see here, if you compare this and this figure, the archenteron size is getting bigger and bigger, whereas the size of the blastocell, it is decreasing day by day, and finally it will get disappear. So when you reach the final event of gastrulation, then what will happen is there will be no more blastocell. Only the archenteron is visible here, surrounded by endoderm, that is formed by the megameres. So here you can see the three layers, germinal layers: ectoderm, this ectoderm, then mesoderm, and then endoderm. So there are three layers. So this stage is known as gastrula, and the process of formation of gastrula is known as gastrulation. So how these germ layers are, it will form, and let's discuss about that. So as I told you before, when the gastrulation stage is completed, then there will be the three layer you can see clearly. The outer layer of micromeres that forms ectoderm, that I already told already, and inner layer of megameres from the endoderm, and the chorda mesoderm that is formed by the fusion of the fusion of mesoderm as well as the notochord that forms the mesoderm in between ectoderm and endoderm. Now, it may be asked in exam, right, the significance of gastrulation or importance of gastrulation. So this gastrulation, as I told before, it is very much important stage of embryogenesis. So the first thing that it is really important is that it has formed the three germ layers, which is very important because these three germ layers form the different organ of the body. Similarly, it will form the body axis, anterior, posterior part, because of rotation of the embryo. And here there will be the movement and movement of cells, rearrangement of cells, and it will form the different structures within the gastrula. Similarly, it will start the development of organ because these three germs layer will start forming the different organs that we'll discuss in the next coming up topic. And it also forms the notochord, which will turn into vertebral column in future, and neural tube will also form here, and that will in future turns into spinal cord and brain. Similarly, the cell which is present here, they will start forming the different organs based on in which layer it presents. Now the last stage of embryogenesis is organogenesis or organogeny. So it is the process of formation of organs from three different germinal layers: ectoderm, mesoderm, and endoderm. So it occurs by following three stages: neurulation, notogenesis, and formation of xylem. So first process is known as neurulation or neurogenesis. It is the process of formation of um, neural tube or nerve cord, or you can also say spinal cord, and that spinal cord along with the brain. And it is formed by the ectoderm that is developed in the gastrulation phase. The ectodermal cells, which is present as a neural plate, the cells become thick. You can see the cells become thick here, and this neural plate will later turns into the neural tube or nerve cord. Now, in further growth, what will happen? The middle area of the neural plate, it undergoes depression, and the depression will is known as neural group. And this neural group, when the neural group is formed, then on the lateral side, there are two lateral sides. It will come more closer because of deepening of the more neural group. So finally, from the both side, the neural fold, it will finally join together and form a tube-like structure known as neural tube. So this neural tube, uh, it is uh, it is open from the either side for a short time. After that, it will be closed. So anterior white part of the neural tube that will turns into brain, whereas the posterior region develops into spinal cord. The hollow cavity that is present inside the neural tube that you can see here, this is called neurocell, and that neurocell is later filled up with cerebrospinal fluid, and this neurocell will it is the one which will form the cavity of brain and spinal cord, and it is also filled with the fluid known as cerebrospinal fluid. And whenever the neural tube is formed, then as I told before, the cells of neural plate get thickened, right? Here get thickened, but some of the cells nearby neural plate, they do not change, and they left below the neural fold. Here you can see they left below the neural fold, and these special cells are known as neural crest. And this neural crest later on forms the knobs of peripheral nervous system and ganglia of autonomic nervous system. The next is notogenesis or notochord formation. So notochord means it is the future backbone. So notogenesis means it is the process of formation of notochord, and it will always form from the mesoderm of embryo. The mesoderm cells, which is lying in the caudal, the mesodermal cells that is lying in the mid dorsal region, just below the neural plate. So it will remain thick, okay, and that is thicker cells are known as chorda cells, and these thicker cells, it will form these cells, it will become rod-like structure, and this rod-like structure along with the median line, it will form the notochord in future. And these chorda cells which are lying around the notochord, they become vacuolated cells, and they form the meninges of brain and spinal cord. Meninges means is the covering of brain and spinal cord. Now last is formation of cilium. So cilium, it is the body cavity that always lie between the body wall and gut, and it is also mesodermal in origin. Now mesodermal layer, what will happen is it will broken into three pieces. The upper part is known as epimere. The middle part is uh, known as mesomere, and the lower part is known as hypomere. So the mesoderm is divided into three regions. The upper part is known as epimere, so it lies on both sides of the notochord, and the middle part is known as mesomere, and the posterior part that is U-shaped is known as hypomere. So epimere again divides into three more layers. So outer layer is known as dermatome, and dermatome is responsible to form the skin in future. The middle area is known as myotome, so myotome is responsible to form the muscles, and the inner layer that is known as sclerotome, it is responsible to form the skeleton around notochord. Notochord is lying here, so around notochord it will form the skeleton. Now the middle, this is about the outer layer, epimere. The middle layer, that is middle piece, that is called mesoderm. So mesoderm later on modify into the kidney or excretory system. Now let's go to the U-shaped area that is known as hypomere. So the upper part of hypomere, it splits and form the space, and this space uh, later turn into cilium. So when it split, then it is from two layers. The outer layer is known as parietal layer, and the inner layer is known as uh, basal layer. So gap between these two layers, that space is known as splank splanchnocele. So splanchnocele, it will grow in size from either side will grow, and then finally it will fuse together and form the U-shaped cavity, and this U-shaped cavity is known as xylem. So with this, we completed the embryogenesis of frog. So I know it seems a bit long lecture, but if you repeat this video again, then you can understand. So this much for today. Thank you so much.